it's Mr. Peterson. I'm not in school today, and these are the directions for the work that you're going to be doing today. So, the investigative question I want you to think about in your work today is how do plant and animal cells compare in proportion to the time spent in each stage of the cell cycle? So, a while ago, when you worked with those micrographs A, B, and C the last time I was gone, you figured out which one of the phases of the cell cycle was the longest. But does that vary from plant and animal cells? What are the differences between plant and animal cells that might influence that? So that's the investigative question you're going to try to answer today. So you're going to be looking at some slides. I'll talk about that shortly. But basically, it looks like this. On the left-hand side, you have an onion root. And on the right-hand side, you have a whitefish blastula. That's a word that's probably new to you. You can imagine a root tip, and we've talked about that, about how cells are growing and pushing down, looking for nutrients or whatever. But a whitefish blastula is a ball of cells that when it continues to divide and turn into different types of cells, it eventually would turn into some type of fish. In this case, a whitefish. So on the left-hand side, we've got the onion root tip. You can see those rectangularly shaped rows of cells that we find in plants. And on the right-hand side, we have a whitefish blastula, which is a ball of cells. Now, this ball of cells has been sliced, so we're only looking at a section of it. And it looks kind of like a pizza, and you can see the individual cells. They're irregularly shaped. Some of them you can see the nucleus, and some of them you can't. Those are magnified at 150 times, both of these images. So if you look at that, you can make a comparison as far as which cells are bigger in the whitefish or the onion. So the whitefish cells are actually quite large compared to the onion cells. So hopefully that will help you identify the stages of the cell cycle: interphase, prophase, metaphase, anaphase, or telophase. Now, some resources that you have available to you. Number one, you have prepared slides. So those are slides that already have material on them. They're labeled. These two that I have pictures of frog skin and frog blood smears. Well, you're not looking at that. You'll be looking at mitosis. It might say allium, which is the scientific name for onions, and it'll say whitefish blastulas. So those cells are already prepared. And you have a microscope. You know how to use that to make some observations of those prepared slides to help you decide what cells look like what. In Google Classroom, there's an atlas of images, and I'll show you where that's at in just a second. But it has a bunch of pictures. You've seen images like that already. A bunch of pictures of plant and animal cells in the various stages of mitosis. And then I also have an antique. It's called the National Teaching Aids Slide Viewer, and it kind of looks like a microscope, but it's much simpler. So the National Teaching Aids Slide Viewer, you get a strip of images that look like the picture that I'm showing you right there in the middle, and it slides through the slide viewer. You don't need to plug it in or anything. It has that white background that acts as the light, and it helps you make some observations. So those are all things to help you to answer that question about the difference between plant and animal cell mitosis. So some things I need you to think about with your group before you begin. Number one, what type of data are you going to collect? So you already have a little bit of experience with regards to counting cells in various stages of mitosis and the cell cycle. You've already done a cell cycle counting activity. How are you going to collect that data? So the second question, how are you going to collect the data? And number three, how are you going to analyze the data? What's that going to look like? What's the analysis of that data going to look like? So you and your group need to think about those questions. There are some other kind of additional questions maybe to help frame that up, and you'll find those on the directions for the lab. So on Google Classroom, you're going to see a box called Plant and Animal Cell Cycle Lab, and there are 3 things that are available to you. Number 1, the directions. Number 2, over on the right-hand side, the Mitosis Atlas PDF. You don't have to download it. You should just be able to open it and use it that way. If you want to download it, then you got to go through the 3 dots in the upper right-hand corner and then download it, open it in a new window, and then download it that way. Or you can just use it in the Drive window. You will have a hard copy of the directions. And then the preliminary slides, those are the ones that I'm using for giving you directions. So the directions look like this. So here in figure A and B, just make some observations. What do you notice about those cells in A, which are plant cells, and B, which are animal cells? What are some similarities and what are some differences? And how might that play into what could be a difference in the cell cycle? There's just the list of things we've already talked about. And then here are those 3 questions that are on the slideshow with some other questions to help you think about that. The last page here, there are some hints about using the resources that you have available. The atlas looks like this. So it's some colored pictures, right? So there's that image from the slideshow. And then there's some other pictures that show interphase, prophase. Now here it says early and late prophase. I don't care so much about that. But you can see that definitely you can see chromosomes starting to show up in prophase. And then here's metaphase, anaphase, and so on. The next set, here's that squashed blastula, the pizza with all of those cells. And then zoomed in at, this is 1,000 magnification, so this is really close. One of the things that you'll notice when you look at the blastula on the slides is there are going to be little circles, little discs. There's going to be several of them across the slide for you to look at. It's going to require some patience and some work in adjusting the light on those microscopes to get a really good look and a really good image to be able to answer that question and count some cells. Sometimes the stain is a little bit faint, so just be patient with that. So the question, again, if we go back to this question, how do plant and animal cells compare in proportion to the time spent in each stage of the cell cycle? That's what you're trying to answer today, using those resources that are available. Again, I'm not there today. I know you'll be respectful and you'll do what you're supposed to do. You're good, independent workers. Thanks. A couple of things. When the hour's over, make sure you put the slides back in the correct trays, that the onion slides are with the onion slides and the whitefish slides are with the whitefish slides. Everything is labeled. Make sure the microscopes are turned off. Be careful with those national slide viewers. They're just fragile. They're old and well loved. They've been used by generations of students have been on San Margaret. So make sure you pick up and put things away. I'm not asking you to do more than collect some data and perhaps analyze that data, and you know what that looks like. So you and your group are going to have to come up with a way to do that, and then eventually come up with a claim. Now, right now, I'm not asking you to do a claim, evidence, and reasoning, but that's kind of the direction we're heading. We'll probably get to that next week.